All right, we're going to finish up chapter 7 today. We're talking about the membranes and cells. And remember, in the phospholipid layer, that bilayer, there are proteins, and they have different functions, and one of them is called a carrier protein. What happens is a molecule binds to it, changes its shape, and then it shuffles it across the membrane. It's kind of like enzymes, you know, they have an active site that they join, and then they change the shape and your products are different. It's kind of along those same lines, okay? And transport proteins are very specific, okay? There's not like one protein that can move everything. There's probably, you know, if 20 things need to be moved in, there's 20 different proteins to do that. So they're very specific to what they move in, okay? Whether it be another enzyme or protein, a hormone, ions, whatever needs to be, they're very specific for what they move in. So there's a lot of different proteins in those membranes, okay? Now, when they move stuff, they move it a couple different ways, okay? There's passive transport. It's just like diffusion. And diffusion is a natural thing, doesn't require energy. Diffusion is molecules tend to spread out evenly, okay? If you put a bottle of perfume in a corner of a room, pretty soon the whole room will smell like that perfume because those molecules want to spread out as far as they can. So that's a natural occurring thing. It doesn't take energy for it to happen, okay? Although each molecule moves randomly, for some reason they do just like to spread out as far as possible, okay? So the diffusion of the molecules, kind of directional. It usually goes from low concentration to high concentration as far as the water. But if you're talking about the molecules themselves, if you have a high concentration of molecules, they're going to spread out so it's all even. So they really actually go from high to low. So it's opposite when you think about water. And we'll talk about that in a minute, okay? All right. Actually, on this slide we will. Okay, osmosis is like diffusion, but it's just water, okay? When you talk about osmosis, you're talking about the movement of water, okay? It's water will move across, they call a selectively permeable membrane. Not everything can move across it, but water can always move across it, okay? And really what the water does is it moves to diffuse things to, you know, if you have high concentration in one part and low on the other, the water is going to move toward the high to like dilute it because it wants it to be the same on both sides. So the amount of water is going to be different, but the particles are going to be the same, okay? So water diffuses across the membrane from a region of low solute concentration. And again, it's doing that to dilute it. It wants to make sure that both sides, you know, you have your membrane here and you have stuff over here and stuff over here. It wants both sides to have the same amount of stuff, the concentration to be the same. So it's going to move the water. So the water won't be the same. Where you have that high concentration of solute particles, the water is actually going to move into that area so you can have more water on that side. But the concentration of the solute particles is going to be the same. Okay, here you can see, here we have sugar. You can see we have more sugar on this side than we do on that side. And then we have our membrane right here. Okay, so natural thing is water is going to say, hey, we got to dilute this baby. We want the same on both sides. So what we're going to do is the water molecules are going to move across, okay? And they move both ways, but there's going to be more water coming this way than that way. So what is happening is now you can see they're kind of evenly spread out, but there's more water on this side, okay? That's how it makes the concentrations the same on both sides. So water will move through osmosis to make sure that the concentration is even on both sides, okay? That's called osmosis. Osmosis is like diffusion, but it's only water we're talking about, okay? So water balance of cells without cell walls, and that would be us, mammals, you know. If we don't have that, we can end up with some problems, okay? We can end up with water, the cell gaining or losing water, and it can lead to problems with the cell. Okay, there's one solution that we want. That's called an isotonic. Okay, an isotonic means the solid concentration is the same. If you ever have had to have an IV, they give you isotonic solution. They give you a 0.9 salt solution, and a 0.9 salt solution is the same as our concentration. That's what the salt solution is in our blood. So when we get an isotonic solution, we're getting fluids. You don't just get water. That wouldn't work. So you need those particles. So isotonic is the solid concentration is the same as inside the cell. And again, if you're talking about us, it'd be a 0.9% salt solution, or they call them saline solutions, okay? If we get solute concentration is greater than inside the cell. So you've got more concentration on the outside than in the cell. What's going to happen is water is going to diffuse out to try to dilute that concentration. And what's going to happen to the cell is the cell is going to dehydrate. It's losing its water. So those are called hypertonic solutions. And then we've got just the opposite. If we've got more concentration, the solute particles are greater inside the cell, then water from the outside is going to diffuse in to try to dilute it again, and the cell is going to gain water. In this case, our cell is going to get really tiny. It's going to dehydrate. Here, it's going to plump right up, and it might just even, blow. Here's what we want. Okay, so again, when they give you solutions, if you need to get, when you go in the hospital and get IV solutions, you get an isotonic because you don't want any of this to happen to your cells. Okay, here's what happens. If it's hypotonic, okay, what's going to happen is there's the concentration out here is less than the concentration in here. So water's going to move into, it wants to be the same. And so it's going to get too big and the cell's going to go kaboom. And it's going to, what they call lyse. It's going to explode. Here's normal. They're the same. The movement in and out are the same. Okay, so that works great. That's what we want. Now here, in this case, we have a hypertonic. In other words, we have a lot of solute particles out here and not so much in the cell. So the water is going to move out of the cell out here to take care of that, to dilute it, and it's going to shrivel it up. It's going to dehydrate it because it's losing water. Okay, so these are the different things that can happen with the movement of water called osmosis. Okay, they show it down here too. And these vacuoles, we talked about those before last chapter, they kind of help with that. They try to hold some of the water. Here you can see there's a lot going on there. Actually, it's kind of hard to tell the difference between these two, isn't it? But the vacuole kind of tries to help with that water concentration. Okay, so hypertonic and hypotonic create a problem. Okay, you don't want to be in either of those situations. You don't want your cells to blow up. You don't want your cells to shrivel up. You want to be in isotonic. Okay, so osmoregulation, the regulation of osmosis in your body is necessary. We have systems, we have stuff that helps us do that. Okay, it's a necessary adaptation. So because we can do that, then our cells don't, go kaplooey or shrivel up to nothing. So we need that system in our body. And again, it's to help to control the flow of water. We want to be in an isotonic situation. Okay. There's a protista. It's just a little one-celled organism called a paramecium. And it's hypertonic to its pond environment. So it has contractile vacuoles. And I talked about that a little bit to help, regulate that water. Because again, if you get too much inside, the cell's going to go kaplooey. So it has a little pump, kind of like a sump pump. When you get too much water, you pump it out. That's kind of what the contractile vacuole does in a paramecium. Okay, you can see it right there. So it sends out, it absorbs excess water, and then it has these little things to help get rid of that water so it doesn't blow. Okay, so we have water balance in the cells with cell walls. These would be plants, and they're going to have more of an issue of that because they need water, to make food for us. So they have some other things to help. And again, if a plant cell is in a hypotonic, hypo meaning low concentration, then again, you're going to get this swelling. Where the wall is going to oppose the uptake. This cell is going to get very turgid. And sometimes you see this if you have a plant that you haven't watered for a while. You ever notice it's all wilted? And then you put water on it and then it plumps back up again. Okay, that's what happens in those cell walls. Okay. So if a plant cell and the surrounding are isotonic, there's no net movement. The cell will become limp if it was okay, but there's no movement going on. Because really in a plant, you need to have movement of water. So isotonic is kind of a bummer for them because they want it to be moving. So they use hypo and hypertonic solutions to help them. Okay. All right. Then sometimes we have diffusion against the gradient. Okay. Natural diffusion is from high concentration to low concentration. For water, for osmosis, the water is going to do what it can to make sure the concentration's on the same on both sides. So water is actually going to go into your, to make sure that your high concentration, it dilutes it, okay? But sometimes we have to do it the other way. Sometimes those transport proteins in the membranes, they have to speed those molecules across, and they can't just wait on diffusion. They gotta speed her up, okay? And in order to do that, they have to actually use energy, okay? A facilitated diffusion is opposite of the natural occurring thing, okay? So a facilitated diffusion requires energy, all right? And they can call it active transport also, okay? So when you have a facilitated diffusion, if it's passive, you're not going to use energy. If you need it to be active, if you want to move solid particles against the gradient, in other words, you have a high concentration and you're going to put more on the high concentration. That's not normal, okay? But that does happen, okay? So if that happens, sometimes what you have to do is, and again, it's the situation, what's going on. I mean, there's more sugar in the mitochondria and we're going to put more sugar in there. Well, that's not natural if you go by the regular. So sometimes you have to move solutes against the concentration gradient. Okay? All right. The active transport moves molecules against, again, they need energy in order for them to move, because they're moving opposite the natural way. Okay? Natural is everything to be even. But if we need to have even more over here, you know, and we got even more coming in here. So instead of diluting it, we need to even concentrate it more. We have to have energy. And the energy is ATP. ATP is made in the mitochondria, so it's right there, right inside the cell. But they need that high energy phosphate bond in order to do this. Okay? There's only certain proteins that'll do that. And again, they're embedded in the membrane that help with this. So most of the time, you just, you know, they help things move across just using natural diffusion. But sometimes they need a little energy boost, and to move maybe bigger things, or, you know, some things going against the gradient, you're going to have to use energy, and that's called active transport. Okay. When you have a lot of transport going on, sometimes they'll chomp things up. They have different things called exocytosis or endocytosis. Okay. So if you have small molecules and water and they want to enter or leave the cell through that layer, they can do it through the proteins. Okay, that's a natural thing. But what happens when you get large molecules like polysaccharides, starch, glycogen, you know, those big ones, big proteins? If they need to cross that membrane, they're just not going to squeeze through there. It's not going to happen. So they have to have vesicles. They have little tiny compartments built for them that they travel in. Okay, and if you do a lot of it, it's going to take energy. Okay, so vesicles is made in the cell, and there's little particles. I showed you with the Golgi apparatus. You know, they dump their product in there and they put it in like a little container, and then the container's taken to the end of the plasma membrane, and it opens it up, and boom, it's released into the bloodstream. Okay, exocytosis. There's endo and exo. Exocytosis transports vesicles to the membrane. They fuse with it, and then they release their contents outside the cell. So exo means outside, okay? So these are stuff coming inside, made inside the cell, and then releasing the outside, okay? A lot of the secretory cells, like if they're making hormones or, you know, stuff like that, they're going to export their products, okay? So exocytosis is to export, to take stuff that's made in the cell and release it on the outside, okay? Endocytosis is just the opposite, where you're going to take in macromolecules. They're going to form a vesicle at the plasma membrane. They're going to make a little, you know, container. They're going to throw that stuff in there, and then it's going to be transported inside the cell. So it's endocytosis, okay? It's the reverse of it. So, and again, you can only do it with certain proteins, but endocytosis is taking on the inside, okay? Taking it in the cell. And there's actually 3 types. I'm getting toward the end here, you guys, and I really want to finish. So there's phagocytosis, where they actually just chomp it up like an amoeba. There's pinocytosis. It's kind of like a little straw, and they suck it in. And then there's receptor mediate, and that's where you're going to get your proteins that have certain active sites that suck it in. So when you're doing endocytosis, you have 3 different ways that you can actually incorporate it into the cell. Okay, and here's a picture of it. Let me see. Yep, okay. Oh, I shouldn't have done that, you guys. I'm just about done. Anyway, there's a picture there. Oh dear, I'm sorry. There's a picture there that shows you the diagram, and then there's one more slide. about it, and just read through those because I'm going to run out of time. So